Hey all, we will be looking at B3.1, which is gas exchange in this video, and we want to be thinking about how do mammals exchange carbon dioxide and oxygen. So we're going to be looking at pages 256 to 61, and we want to be thinking about the structure of gas exchange surfaces and how they contribute to the function of gas exchange. So firstly, gas exchange organism needs to move carbon dioxide and oxygen. So one needs to go in, one needs to go out. And terrestrial organisms do this via air, while aquatic organisms do this via water. Larger organisms need specialized surfaces, but with smaller organisms, they can sometimes use just their surface or sometimes skin to have this exchange facilitated. Surfaces, though, do need to be permeable so the gases can diffuse freely, have a large surface area to volume ratio, be moist. In terrestrial organisms, this is especially important. So gases can dissolve, and this is why the inside of our lungs is moist. And then thin. So they have a very thin cell lining. Usually it's about 1 cell thick, so that they have a short diffusion pathway. In order for gas exchange to happen, diffusion needs to be occurring, and that can only happen with a concentration gradient. So aerobic respiration, that's mitochondria using oxygen to make CO2 and ATP. In small organisms, it's only the mitochondria that are maintaining the concentration gradient. But in bigger organisms, capillaries are always picking up and dropping off gases, and that's how a concentration gradient is maintained, as well as tissues respiring, so they're producing CO2, that's going to have high concentration of CO2, and lungs breathing in oxygen, so that's going to have a high concentration of oxygen. Ventilation is the inhale-exhale process, and if you're a terrestrial organism, you're breathing in and out in the air, but if you are an aquatic organism, you're removing gases from the water by running it over the gills. So you can see gills here, they have capillaries here. So the carbon dioxide is going to diffuse out into the water, and oxygen is going to diffuse in. So as a byproduct of that, organisms need to have a fair amount of oxygen in their water so that they will be able to get enough oxygen to sustain their bodies. So ventilation in mammals, this breathing process, bronchi and trachea have cartilage to keep them open and smooth muscle fibers. So if you touch your throat right here, you can feel bumps. This is your trachea, and this is the bumps you're feeling are the cartilage. And so that's this here. And then the bronchi are these two branching tree-like structures that are going to turn into bronchioles, and then eventually into an alveoli, which are these little air sacs down here. In order for us to breathe, we need to have antagonistic muscles, so muscles that are operating in both contractory, so they pull in and then they also pull out. And in this case, we are looking at our internal and external intercostal muscles, which are muscles between the ribs. And those muscles, external intercostal muscles, are going to pull externally up and out, and internal intercostals will pull down and in. And the movement of these muscles combined with the movement of the diaphragm, which is also a muscle, will cause the pressure to change in your lungs, causing air to move in or out. So when you breathe in, what's happening is your muscles in your ribs lift up and your diaphragm flattens. And so the volume inside your lung cavity there becomes quite big, and the air goes in because the pressure drops. And then the opposite is true for breathing out. The muscles pull down, those are the internal intercostal muscles, and then the diaphragm relaxes, which allows it to dome up. So it's going to reduce the volume and increase the pressure, and the air will push out. So we talked about the little air sacs inside our lungs. These are called alveoli, and they're surrounded by a dense capillary network and a single layer of cells. So we have these epithelial cells going around. It's 1 cell thick, and that's the diffusion process pathway. And then around here, there's many capillaries. And we have many alveoli in our bodies, and those alveoli actually altogether make it's something like 30 million. They're going to make up more than 40 times the surface area, if you laid them all out, of our whole skin. So it's quite a lot of surface area to volume ratio. And there are two types of main cells in the alveoli. They're sometimes called AT1 cells, or these are type 1 pneumocytes, and they allow for gas exchange to happen in the alveoli. So these are the type 1 cells. They're very, very thin. And then type 2 cells produce surfactant, and surfactant is going to prevent the alveoli surfaces from sticking to themselves and then collapsing. And these are more ovoid shape. You can't really see them in here, but what's going on with these are the phospholipid tails, and they're pointing into the alveoli. So they're preventing the sticking together based on these phospholipid tails that are being made. And so you could imagine that this would be a type 2 pneumocyte here. We will see in different videos, oh, there's a type 2 cell. So much bigger, and it's producing that surfactant. Alveoli specialization. So again, type 1 pneumocytes, AT1 cells, these are going to allow for passive diffusion. There's very few mitochondria, but the cytoplasm is very small. They have thin walls with capillaries nearby. And then type 2 pneumocytes, there's more of these than type 1, 90% of the cells, but only 5% of the surface area. And they have a dense cytoplasm with many organelles to make phospholipids that's stored in vesicles and secreted via exocytosis. So you can imagine that if they're making phospholipids, they would need many organelles to make that happen. Alveoli are lined with moisture to allow carbon dioxide and oxygen to diffuse, and those phospholipid tails face into the alveoli, and proteins from the lamellar bodies are interspersed. The two together reduces surface tension and prevents alveolar collapse. So if there are proteins in the vesicle type structure, they are going to work with these phospholipid tails to prevent the alveoli from collapsing. So if we wanted to measure lung volume, this is page 261, there's a few different things that we could be measuring, and we would use something called a spirometer to measure it, where you breathe into it. First one is tidal volume. That's how much air is going in and out of your lungs as you breathe. So you can see there, that's tidal volume. Ventilation rate, that would be how many times you're breathing per minute. Vital capacity is the total volume of air exhaled after a maximum exhalation. After a maximum inhalation, or the total volume of air that can be inhaled after a maximum exhalation. And inspiratory reserve volume, so that's the amount of air you can inhale after inhaling normally. So you breathe in, and then you have to breathe in all the way. That would be this peak here. And then the expiratory reserve volume would be the same thing. You breathe out normally and then huff everything else out that you can. So all of these different things are measuring different components of your lung structure and healthiness, but there are different factors that could be measured for lung volumes. So to summarize, diffusion occurs because of a concentration gradient. Surfaces need to be thin, moist, permeable, and have a high surface area to volume ratio. Antagonistic muscles, internal and external intercostal muscles, are responsible for breathing, along with the diaphragm. And they cause pressure differences in the lungs to make that happen. Type 1 and type 2 pneumocytes allow diffusion of gases in the alveoli. And well, actually, type 1 pneumocytes allow diffusion of gases, and type 2 pneumocytes produce surfactants to prevent the collapse of alveoli. And a variety of measurements can be made for lung function that we just saw on the last slide. Thank you for watching this video. I hope you were able to understand most of the information.